And I'm going to call our keynote speaker to the podium, Dr. Philip Lupo. He will deliver the keynote on cancer risk in children with birth defects, how Kids First can move observation into understanding. Dr. Philip Lupo is the director of the Epidemiology and Population Science program in the Texas Children Cancer and Hematology Center. He is a Kids First principal investigator, and his area of research is cancer risk in children with birth defects. Dr. Philip Lupo, I would love to invite you to join me and share your experiences.
Thank you very much, Marcia. It was quite an introduction, as well as I really enjoyed hearing your background setting up the story of Kids First. So bear with me while I pull up my screen. And I trust you can see everything okay. All right. So again, thanks again for that introduction, Marcia. I am really honored and privileged to talk a bit about our research and how Kids First has really moved that area of study forward over the past few years. So I was actually at that meeting in, excuse me, 2012 that the NIH sponsored, trying to address this question of cancer risk in children with birth defects. And that really set the stage for a lot of the work that I'm going to be talking about today. And just to get started in terms of an outline, I'm going to share some of the observations that we've been able to generate through our registry-based studies, which I think has really set the stage for the sequencing projects that have been selected as Kids First cohorts. And I'm actually going to highlight two of those studies that have been selected for X01 funding through Kids First. The first of which is our GOBACK, or genetic overlap between anomalies in cancer and kids study, where we're really trying to identify novel congenital anomaly cancer predisposition syndromes. And the second study that I'm going to highlight is our Down syndrome acute lymphoblastic leukemia study, which I think will hopefully point to the utility of Kids First in identifying those children with congenital anomalies who are more or less likely to develop cancer. So as Marcia said at the outset, I think there's actually quite a bit that we know about birth defects and cancer risk. And I've been using this illustration based on a March of Dimes categorization of birth defects for the last few years, but I think it's really important. So, I mean, obviously, when we think about birth defects, you can categorize them in multiple ways. And I find this designation by the March of Dimes to be particularly useful in thinking about cancer risk. So one of the most important groups of birth defects that we think about are those that are driven by chromosomal anomalies. And again, we do know through decades of evidence that children with certain chromosomal anomalies are more likely to develop cancer. And probably one of the best examples or best known examples of that would be trisomy 21 and acute lymphoblastic leukemia or acute myeloid leukemia. There's also a group of defects that are known to be due to single gene defects or Mendelian syndromes. And again, I think a good example of that would be something like Costello syndrome, which is characterized by dysmorphic features, congenital heart disease, and other birth defects, and the subsequent risk of certain malignancies like rhabdomyosarcoma. But really the largest grouping of birth defects really fall into this multifactorial or what some people call non-syndromic group of birth defects. And I think this is where we know less about cancer risk, or at least we did know less about cancer risk, but where studies over the last decade have really moved our understanding. So what are some research questions related to this, and how can Kids First improve our understanding? So obviously, one of the most important questions, I think, in terms of understanding biology and thinking about surveillance and prevention would be, what are the birth defects that are associated with specific cancers? And then secondly, could some of these associations represent undiscovered Mendelian syndromes? And then importantly, why do some children with birth defects develop cancer while others do not? And can we leverage this information again to think about treatment and or surveillance strategies for these individuals? So that really led to a multi-state project that is actually ongoing still, but started with 4 states, as you can see here from this slide, including Texas, Michigan, and Arkansas, as well as North Carolina, where we linked birth defects registries with cancer registries in these locales to generate a birth cohort of a little over 10 million children. And with that cohort, we were able to identify 4 groups of individuals where we could really dig into cancer risk among children with and without birth defects. And I'd like to point out Jeremy Schraw, who was originally a postdoc in my group, but is now an assistant professor here at Baylor College of Medicine, did a great deal of work in harmonizing and assembling these data for analyses. And again, the 4 important groups are outlined here in these boxes. But you can see, even with a cohort of 10 million individuals, that we had a little over 2,000 children who were born with a congenital anomaly or a birth defect who went on to develop cancer. So one of the largest assessments of its kind. And some of the 10,000-foot observations from that analysis were consistent with some smaller and older reports, but I think notable nonetheless, and as you can see, relatively precise with our sample size. So what we observed is that children with non-chromosomal birth defects were about 2.6 times more likely to develop cancer compared to children without those birth defects. Again, this is pretty consistent with previous studies evaluating non-chromosomal birth defects and cancer risk. And then children with chromosomal anomalies were about 12 times more likely to develop cancer compared to children without such chromosomal anomalies. And then moving on to specific birth defects and specific cancers, we did note some interesting observations that I think will lead to, I hope, findings from the Kids First program and populations that have been included as part of Kids First, which I think could really be explored to understand these associations. So the first of which is that we noticed embryonal tumors were associated with a range of birth defects. And this was particularly true for hepatoblastoma and neuroblastoma. And again, you'll know that neuroblastoma is included as one of the Kids First cohorts. Secondly, we saw the same pattern of associations across germ cell tumors, another cohort that's been included as part of Kids First sequencing. And then moving from specific cancers to specific birth defects, we did notice that children born with CNS defects were at an elevated risk of developing certain brain tumors, which I think is particularly interesting if you think about common developmental pathways that might have been disrupted at key points during gestation. Secondarily, we noticed that children with oral clefts actually did not have the same cancer risk compared to children with other defects. And I think this is an area worth exploring, especially in light of the number of oral cleft cohorts that have been included as part of Kids First. And then digging down deeper into those seemingly non-syndromic birth defects that were associated with cancer, again, because of our sample size, we were able to look at a number of specific associations that had previously been excluded in analyses that were published. And notably, among those 72 associations, 40 were significant after correcting for multiple comparisons. So I think this points to the importance of birth defects and cancer risk and the role of disrupted development on cancer in children. So a few specific associations among children with seemingly non-syndromic birth defects that I'd like to point out are listed here. Specifically, children with certain congenital heart defects were more likely to go on to develop certain embryonal tumors like hepatoblastoma and neuroblastoma. Again, I think this points to the utility of Kids First in that there are cohorts that have been sequenced of children both with congenital heart disease as well as neuroblastoma. So thinking about the common pathways that underlie these phenotypes, I think is a rich area to explore with those existing data. And then there were other associations that hadn't been previously reported that we were particularly interested in. And I'm going to draw your attention to the association between choanal atresia, which is a type of craniofacial defect, and acute leukemia. So in this case, children with choanal atresia were about 10 times more likely to develop acute leukemia, which was particularly interesting to us because it wasn't an observation that had been made, at least one that we noticed in the literature, and not something that's associated with classical cancer predisposition syndromes. The other observation from the study, and I think this points to the challenge in trying to call birth defects non-syndromic, is that we noticed with increasing number of seemingly non-syndromic birth defects that the risk of cancer increased as well. So you can see here from this cumulative incidence plot that with each increasing number of major non-syndromic birth defects, the risk of cancer increased significantly, whereby children with 4 or more defects were about 6 times more likely to develop childhood cancer compared to their peers without birth defects. And again, these are children that did not have a syndromic diagnosis, at least in the birth defects registry data that we had access to. And again, what I think this points to is likely that there are undiscovered syndromes within these data that could yield insights about cancer predisposition and about development in general. So moving on from those registry observations into trying to understand what's driving these associations has really been the impetus of the enrollment portion of our study, or the GOBACK family cohort. And Dr. Sharon Plon and Dr. Annika Sabo have been amazing partners with me in this endeavor. And aside from the work that we've done in linking those registries, we've also used those registries to identify families where the proband has both a congenital anomaly as well as a cancer diagnosis to conduct whole genome sequencing, both on those individuals as well as family members. And I'm going to highlight one family in particular here that I think is indicative of how we can use kids first to really get a better sense of what's driving these associations and what might be important for thinking about novel syndromes. So in this one family, as you can see here, which has really been led by Samia Sasudia, who's a PhD student in Dr. Plon's lab, we observed about a 5 kilobase exon deletion in USP9X. And this is obviously not present in either parent or the proband's sister. This was validated in a PCR experiment. But what's interesting about the phenotype of this proband is that it really matches nicely, I think, with the registry observations that we had in the GOBACK study. So for instance, the child had multiple structural birth defects and did not have a syndromic diagnosis. If you were to look at this set of birth defects, you might imagine that the child had CHARGE syndrome, for instance, but did not have a CHD7 variant. And also the child had choanal atresia and diagnosis of precursor cell lymphoblastic leukemia. So this association between choanal atresia and acute lymphoblastic leukemia that we saw in our registry data, which led us to enroll this particular family, I think really bore out, I think, some interesting observations in terms of genomics. Because again, remember, this association that we observed was not necessarily one reported previously. And then I think digging into USP9X and its role in both development and cancer, I think points to some of the evidence that Marcia alluded to at the beginning of her presentation, is that these genes really can be implicated in several developmental and cancer pathways. And this is true for USP9X, as you can see from this biorender illustration that Samia prepared. And then Samia has done a great deal of work to identify families that are characterized by these USP9X variants. And to date, there have been 42 identified individuals with variants in USP9X. And they all seem to be associated with a female-specific syndrome that includes both developmental delay and congenital anomalies. And what's even more interesting among these 42 individuals with this novel syndrome is that 3 of them have gone on to develop acute lymphoblastic leukemia. So if we were to look at ALL risk in our USP9X cohort or in this USP9X cohort versus SEER data, you can see there's a significant enrichment in leukemia incidence and risk. Which I think is notable in that this is not, again, a known or a well-characterized leukemia predisposition syndrome. And then if you move from germline sequencing to somatic sequencing of tumors from this pan-cancer analysis that was published several years ago now in Nature by Ma et al., one of the most mutated genes in the context of acute lymphoblastic leukemia somatically was USP9X. So I think this is beginning to lead to a hypothesis that I hope you're drawing, at least through the presentation of these data, that USP9X could be a novel ALL susceptibility gene that's associated with a charge-like syndrome. So again, I think this is, I hope, an example where we move from association in our registry-based studies to genomic sequencing of informative families to identify these alterations that could underlie novel syndromes. So what are our next steps related to this? Again, I mentioned that, that our GOBACK cohort was selected for sequencing as part of Kids First. So we're really hoping now to move beyond, again, these registry-based studies to enroll more families where these children have both a birth defect and a diagnosis of cancer. And through our X01, we're planning to determine the frequency of known cancer predisposition variants among children with congenital anomalies and cancer, and then to identify novel variants that could underlie syndromes that haven't been characterized, like with our USP9X example, and also describe the landscape of somatic alterations in these children. And again, this is an effort that Sharon Plon and Logan Spector and I have been leading over the last 4 years. And it builds largely from the Children's Oncology group. And for those of you who aren't familiar with COG, it's an amazing resource, not only for developing clinical trials for children with cancer, but also for observational studies in these populations. So you can see there are more than 200 research sites throughout the United States that are affiliated with the Children's Oncology group. And about 90% of children diagnosed with cancer in the US are cared for at a COG member site. And because we can leverage COG protocols like Project Every Child or APEC 14B1 to enroll patients into our studies, it gives an even richer platform to study cancer risk in these individuals. And an added component to Project Every Child is a recent questionnaire that was developed and implemented that does ascertain birth defects or structural birth defects at the time of diagnosis. And as you can see here, it's a limited list, but one nonetheless that we can use to identify children enrolled in Project Every Child that have a reported birth defect. And you should know that there are currently 25,000 children that have been recruited to Project Every Child. So again, you can imagine the power behind not only consent to this protocol, but the biological specimen banking that goes along as part of it. And our X01 is really going to focus on all children that are part of Project Every Child that have a malignancy as well as a reported birth defect, as you can see here in this table from our grant application. And as part of that effort, we're going to be sequencing a little over 700 individuals, and that's going to include both their germline and tumor samples that will be included for whole genome sequencing and additional exome and transcriptome sequencing on a subset of the tumors that are banked as part of Project Every Child. But as you can imagine from Project Every Child, this list is very limited in its scope. And one thing that we've been able to build from are the other mechanisms that are part of Kids First, specifically in the RO3 mechanisms to enhance phenotypic efforts in Kids First cohorts. So because this list is relatively limited, we recently obtained RO3 funding to collect extensive phenotypic and clinical data from children with congenital anomalies and cancer that are enrolled in Project Every Child. And then we're working closely with Allison Heath and Adam Resnick to integrate this information into the Kids First Data Resource Center. And I think this is going to be a powerful tool to expand upwards as we think of integrating data from Project Every Child into the DRC, which I think is a goal that a lot of us would like to see come to fruition. So overall, in terms of GOBACK, I think, I hope I've showed that these population-based registries can really be leveraged to inform novel genomic analyses. And what's important, I think, is that birth defects based on these estimates could account for upwards of 10% of childhood cancer. So clearly an important consideration in thinking about the etiologies of these conditions. Of course, the reasons for these associations are complex and probably involve multiple pathways, but I do think in some children, inherited and de novo genetic variants are likely to explain the co-occurrence of both phenotypes. So moving on from our work in GOBACK to Down syndrome associated leukemia in particular, I think most of us know, again, that trisomy 21 is one of the strongest predictors of acute lymphoblastic leukemia, as well as AML. In fact, we've known this for decades. And about 2% of children with ALL and about 10% of individuals with childhood AML have a diagnosis of Down syndrome. But what's also interesting in relation to Down syndrome associated acute lymphoblastic leukemia is that the clinical features are relatively different than among those without Down syndrome. So we see distinctive genetic alterations in the leukemia profile of children diagnosed with Down syndrome, whereby those typical ALL alterations like hyperdiploidy and ETV6-RUNX1 are less common, whereas CRLF2 and JAK alterations are much more common. So about 50% of children with Down syndrome that develop ALL have CRLF2 rearrangements compared to less than 10% in the non-Down population. We also see poorer outcomes in this population. So children with Down syndrome that develop ALL are more likely to relapse, and they're more likely to have treatment-related toxicities and associated mortality. So our research is really trying to understand why children with ALL, or why Down syndrome is such a strong predictor of ALL risk. And we wondered if there are specific germline variants associated with ALL in children with Down syndrome, and how these germline and somatic variants could interact within the context of trisomy 21. So fortunately, there's quite a bit we do know about the genetic architecture of ALL as demonstrated in a range of genome-wide association studies. But the one thing I'd like to draw your attention to in this table is that there are currently, or at least among those top hits that have been identified, there are no variants on chromosome 21. So the idea of a gene dosage effect in chromosome 21 related to ALL is not something that's really borne out, at least with our preliminary understanding and data. So we worked together through COG as well as other populations to conduct a genome-wide association study of ALL susceptibility in children with Down syndrome. And we're able to assemble multiple cohorts to this end. And through that effort, we were able to collect genomic information on a little over 500 children with Down syndrome associated with ALL, and more than 1,000 children with Down syndrome that did not have a report of ALL. And these are results from that publication that was highlighted in Blood about 3 years ago now. But I want to draw your attention to three things in this particular slide. The first of which is that even with a little over 500 cases, we were able to see genome-wide signals. And I think that's notable for those of us who work in genome-wide association studies to see such strong signals. And I think that points to looking at cancer risk in these highly susceptible populations can really yield insights that you may not get in larger populations. The second thing that I want you to note is that several of these effect sizes are relatively strong. Again, maybe not surprisingly. And even though our top hit was in IKZF1, one of our strongest associations observed were in this missense variant in CDKN2A, whereby children with these variants were almost 4 times more likely to develop ALL compared to children without. The next thing I'd like to note is that all of these variants have actually been identified in genome-wide association studies of acute lymphoblastic leukemia in the general population. So while we did see these significant signals, they weren't necessarily unique to Down syndrome. And then the last thing I'd like to point out is that obviously there's an absence of variants on chromosome 21. And that's largely due to the limitations of the SNP array data in trying to understand genetic risk related to those variants in the context of a genome-wide association study. So I think something that's setting us up for the next step in our whole genome sequencing assessments that I'm going to highlight here in a minute. So a couple of observations that I think are key that have really led us to the Kids First program and trying to understand this risk better is that what we did see is that when comparing these known risk variants in Down syndrome associated ALL with kids that did not have Down syndrome, but also developed ALL, is that there were variable allele frequencies even across these populations, whereby certain variants like CDKN2A seem to be more frequent among children with Down syndrome and ALL compared to children without Down syndrome who developed ALL. Whereas other variants like ARID5B were seemingly less frequent. So what's beginning to emerge, I think, from this work is that there's a variable penetrance or that the penetrance of these variants does seem to interact with trisomy 21. And again, our finding on chromosome 9 related to CDKN2A, I think, is particularly notable, and that no matter the ALL phenotype or ALL subtype that was evaluated, we saw a higher frequency of these risk variants in a Down syndrome population compared to a non-Down syndrome population. So suggesting that some of these risk variants are perhaps more frequent, especially in the background of trisomy 21. So setting these children up for a greater risk of ALL susceptibility than a non-Down population. So conclusions from the GWAS, again, that have really led to what we're planning to do as part of Kids First is that the established non-Down syndrome ALL susceptibility loci do seem to contribute to ALL risk in children with Down syndrome. And rather than exhibiting a unique susceptibility locus, trisomy 21 seems to modify the penetrance of ALL susceptibility. And we often saw a greater magnitude effect in the variants that were identified. So our next step is really to take this cohort that we were able to assemble through our GWAS and include it as part of Kids First, and also through the Include project, which Marcia also alluded to in terms of integrating across NIH efforts. And what's notable is that children with Down syndrome not only have this elevated risk of developing acute lymphoblastic leukemia, but they're also at a greater risk of developing certain structural birth defects. So AVSD, or atrioventricular septal defect, is probably one of the best examples of that, whereby children with Down syndrome have a 2,000-fold increased risk of being born with AVSD compared to children without Down syndrome. So this really sets the stage that risk-associated genetic variants in the background of trisomy 21 could lead to a higher penetrance of multiple co-occurring phenotypes in children with Down syndrome. And this includes AVSD and acute lymphoblastic leukemia. And this was the foundation of our XO1, where our two aims are really focused first on identifying genetic variants underlying AVSD in children with Down syndrome, and then taking that larger population and trying to identify variants that underlie ALL risk in children with Down syndrome, with a particular focus on rare structural and chromosome 21 variants, which are mechanisms that we weren't able to fully assess in our GWAS. And in terms of resources, with the DS-ALL sequencing, what I think is a real advantage is that we could include not only germline samples, but also tumor samples on these children, whereby we have a little over 350 paired samples to look at germline somatic interactions in this context. So the studies that are underway are really pointed to doing a comprehensive analysis of heritable variation associated with ALL risk in children with Down syndrome. And as I mentioned, we really want to focus this study on structural rare and variants in chromosome 21. And then the next step, I think, because children with Down syndrome have co-occurring birth defects, is to try to identify the role of co-occurring birth defects in this population on ALL susceptibility. So we do have some primary or preliminary data from our registry linkages that children with Down syndrome that have multiple co-occurring birth defects are more likely to develop acute lymphoblastic leukemia compared to those with fewer birth defects, and that there are specific birth defects that may be predictive of ALL risk. And then linking those children in COG to other relevant data sources to understand outcomes, including early mortality. So I think this is a particularly important area of study because children with Down syndrome and ALL have this distinctive clinical profile that really does predict worse outcomes. So trying to understand the biology that underlies these associations, I think associations are going to be key for better risk stratification as well as treatment strategies. And while Down syndrome does not yet seem to have a unique loci that predispose children to ALL, that they do seem to have a different penetrance pattern whereby these variants do seem to have stronger effects. And I do think it's going to be important, again, through the Kids First program to identify these rare structural and chromosome 21 variants that we can assess through our whole genome sequencing data to understand what proportion of ALL risk is associated with these mechanisms. So I hope that, as part of this today, I've really tried to highlight why Kids First provides, I think, a unique opportunity to explore the overlap between these two phenotypes. And I think related to that, it's easy to see that some of these associations really could be driven by uncharacterized syndromes. And there have been a number of cohorts included in Kids First data that can be leveraged to explore this possibility more than any one group could really tackle. And I think insights into these factors influencing cancer among children with birth defects have multiple implications in terms of how we treat and guide and counsel these individuals. And again, this could lead to improved genetic counseling after genetic testing, surveillance strategies among children with birth defects, as well as treatment interventions. And this effort has just been an amazing collaboration, I think, with individuals that are really countless. And these are a few individuals that have been key to some of the data that I presented today, but the number of individuals I think would go on a presentation much longer than I would have time to present. And with that, I'll end my presentation and thank the organizers again for inviting me to present our work today.